Hello, my name is Arnaud Delorme, and this video is about showing you how to export BIDS data from raw data to BIDS exported data, hopefully showing you that it is very simple to do so and how you can upload your data onto a repository. So here I have some data to play with. And this data is available in the description, so you can download it and you can redo what I did if you want to train to export BIDS data. So I have 4 raw data files from 4 raw subjects, and then I have a file that's called info.docx that contains the description that I will need for BIDS. So if I open this file here, we can see that it starts with the participant IDs. So I have subject 1 to 4, their gender, F or M, female or male, their age, their ethnicity. Here we had Indian, non-Indian, and the fact that the air conditioning was recorded or not during the experiment. This is the description of the task. So the task of the subject was to hear, it was auditory audible task. So they were hearing beep, beep, boop, beep, beep. And so they had to press a button when there was a boop. And there were also other kinds of sound which were relatively rare, like shh, some short noise. So the instruction to the subject: please close your eyes, press the button whenever you hear a high-pitched sound, boop sound, and ignore other sounds. This is another so the description for the experiment that was shown to the subjects. And finally, in the raw data files, we don't have the name of the condition. We have 1, 2, 3, 1, 2, 3, 4, 8. 3 is whenever they press the button, they press the mouse button. Standard is the beep stimuli. Ignore is a stimulus to ignore because it was a glitch. Audible is the boop, and noise is the ch sound. So now that we have that information, we can try to import that data and export it. So here I'm going to first import these 4 data files into EEG Lab. I had a study loaded here, so I'm just going to clear it all. And here import from BioSimi BDI file using BioSig Toolbox. It might ask you to download the toolbox, the BioSig Toolbox, if you don't have it. So I'm going to select the 4 files. And here EEG Lab asks me, do you want to resave these files as .set EEG Lab files? These are the import options. I'm going to leave all the default right here. And here it's importing all the data sets and resaving them as EEG Lab datasets. Now we have our 4 datasets loaded, and we want to export them as BIDS. So before we export them as BIDS, we need to create a study. So we need to create a group of subjects. So create study using all loaded data sets. And here I enter subject 1, subject 2, subject 3, subject 4. I can enter other information, but I'm not going to right here. I don't have specific conditions. The different conditions are in the file, so I'm just going to press OK. So we have a study created, and now that we have a study, we can export to BIDS. So I have the BIDS tool menu right here. If you don't have it, you go to the Manage EEG Lab extension, and then you search for BIDS, and you can install it right here. So export to BIDS. I'm going to leave all the default here. So put in the BIDS export folder. This is the license, changes compared to the previous version, none, and edit task information participant. We'll see about that later, but now we're just going to export the raw data file, see what happens. I press OK. It's telling me you must specify line power. Line power is 50 Hertz. Data was recorded in India. And some data sets need to be resaved. Do you want to resave them now? Yes. So it just created this BIDS export folder, and we need to verify that this we can actually, if we wanted to, we would be able to upload this data set to a standard repository. So that's what we're going to try to do. I'm on Open Neuro right here. So this is where you would upload your data if you're anywhere in the world except Europe, because in Europe you would need a GDPR-compliant server. And for this, I invite you to connect with one of my colleagues. I put his name and information in description. So that's, if you're not in Europe, you can use this website. So here you would press the upload button, and we're not going to do the upload really. We're just going to check if it's compliant. So I select my BIDS export folder, and you see I have 2 warnings here, but no errors. The README file is missing and the offers are missing. But I can continue to upload the data if I want to. This is the metadata, so you would enter the DOI of the paper if you have any, species, so that's human, study type, cross-sectional, it's not a longitudinal study, and then number of trials. I don't need to enter anything here. Here, these are healthy controls. So I'm just going to leave that here. I don't have a DOI for the paper. And then you can continue. It's telling you, you don't have GDPR protection right here. And these 2 checkboxes are if you're using MRI. You want to check this one and not check this one. We don't have MRI, so it doesn't really concern us. Once we press I agree, the data is going to be uploaded to Open Neuro. And once it's uploaded, we can release it. I'm going to show you real quick how to release the data because it's not necessarily intuitive. So here I'm going to go to a dataset which I have already uploaded. So I'm going to go to my datasets, executive functioning data. So once the data is uploaded, you have a page like this one, and it's telling you you need to create a new version of the dataset. So that's what you do here. You add some changes. So it's your first dataset. You would enter initial revision. And if I press add here and create version, is going to create a new version for me. Then it's going to validate the dataset. And once it's validated, you might still have to press the publish button. So you might have to come back and press the publish button, and it's done. Your dataset is published and available to anywhere, anybody in the world. So you can do it like this, and it's going to take you just a couple of minutes to export your data. But of course, that's not the recommended method. Even if you have a very detailed description in the paper of all the events, it's actually better to define the metadata in BIDS. And I'll show you that it's not that complicated. So let's go back to our dataset right here. And now let's go through the other menus. The edit task information, the edit participant information, and edit event information. These are optional, but highly recommended so other people can reuse your data more easily. So the task information, so the dataset name, auditory, oddball, that's an auditory oddball. And then we have to enter a short name, oddball, with no space. That's for the description. Then we have the description of the task. So that's for the README here. We're going to copy and paste for the README. The participant task, we're going to copy the task description and the instruction to participants. The authors, myself, and my student, former student. I don't have a reference to enter right here or institution. The cap manufacturer, I believe it was Easy Cap. The cap model was a 64 channel standard cap. The reference location, it was occipital. These are CMS DRL reference, and they're on the 64 channel cap. They're in the occipital region. There is no ground, so when it's nothing is not relevant, you enter NA, and then here the type of montage 1020. Amplifier was bias MI. The model was active 2. I don't know the serial. It was LabVIEW, but I don't know the version of the software. Hardware filter, that's described on their website, but I'll need to enter it here. Software filter, I didn't have any, so I didn't filter the data. And line noise frequency is 50 Hertz. So I'm done entering all the task information I have available. I'm just going to press OK. Now let's move to the second menu. Let's move to the participant information. So this is how it looks. And here I already have gender and age column. So now I'm going to look at my Word file and just copy that information right here. I'm going to put it a little bit lower. So here gender, it's F, M, age 44, 32, 28, and 35. They don't belong to a group. I don't know if they're Hertz circumference or artifact, but I have additional columns. So I'm just going to enter the ethnicity column, and I'm not going to enter the air conditioning just because that's just an example. Ideally, I would enter it too. But here I enter the ethnicity column and I would enter Indian, non-Indian. I can't have space right here. And then once I'm done doing that, I need to enter some description. Here, see levels, click to specify. So gender, here, biological female, biological male, and then group. I don't have any group, so I don't need to specify anything at the group level, but here I enter of Indian origin for Indian and not of Indian origin. It's self-explanatory here, but it's always good to add additional description. And I'm done. I've described the information for my subjects. I have all the metadata for my subjects. If you have questionnaires, you can add additional columns here. You can also import a text file into here, so you can have additional columns for all the questionnaire they might have entered, might have filled. So I'm done for the task information. Now I'm going to define the event information. And here there's a lot of information. So there's the BIDS field and the EEG lab field. So here it says from sample, value is going to use type, and sample is going to use latency. If I have additional field in EEG lab, I can add also custom BIDS field if I want to. So here, for example, for type, it's good to specify them in more detail. So here, see for type in the file, I have condition 1 to 8. And here I had defined them already, condition 1. So condition 1 is response. Condition 2 is standard. This is ignore error. This is an error of the software. Oddball and noise. And that's it. I'm done with my events. I've described the event. This is not ideal here because the event in the data files are still condition 1 to 8. This is just description of the event. Ideally, we want to rename this event, so we can go in EEG lab and rename the events. But it's better to have the description. It's better than nothing to have the description right here. So I'm going to press OK. And now I'm ready to export the BIDS dataset for a second time. So I'm going to call it export 2. I'm going to leave everything details here. Changes, I can say the changes. I added the metadata compared to version 1. And then I'm going to press OK. You want to resave the subjects? Yes. And now we have BIDS export 2. That looks like BIDS, that looks like the first one, but we have the README file and we have much more metadata now in the second one. Now let's go back to our upload, see if it's still valid. So I'm selecting the folder right here as if I was going to upload it. This dataset has no issue, 0 issue, not even a warning. I can continue, enter the metadata, accept the terms, and upload the data and release it as a second version of this dataset or as a new dataset and delete the other one. Now, I can still do better. I can enter additional metadata using HED tags. And I invite you to look at the videos that describe what is HED, hierarchical event descriptor. These are standard descriptors, standard events for different types of tasks. So different people with different experiments will have the same event. I'm not going to go into detail here. I'm just going to show you how to add these tags. To add the tags, the first thing you want to do is make sure you have the plugin, the CTagger plugin installed. So you type HED, HED tools. So I have it installed here. It's in bold, so I don't need to install it. And then next, you go here, add, edit event tags. So you just add the study, your study, and then continue. And then enter the type. So I want to select type. So here, type 1 was, if I remember well, type 1 was the response. So I just type response. And you see it automatically find the tag participant response. I can add more than one tag. For example, it's an action. So event action. It's also a mouse button press. So that would be this one, mouse button. I'll put a link in the description how you can find your best tag, but typically you can just type them and it's going to find some relevant information. Sometimes it's a bit more complex, like for example, for 2, this is a stimulus. So experimental stimulus. This is auditory stimulus. So sensory attributes, auditory attributes. I could even enter the pitch if I wanted to of the stimulus. I'm not going to do that here. But here, this stimulus is a standard. So if I enter standard, I only have standard deviation. That doesn't work. But after doing a little research, you have expected. So you can just enter expected. And here I can actually copy these. So this is for the standard. I'm not going to enter anything for free because this is just a glitch in the software, presentation software. So these stimuli can be ignored. But here for condition 4, this is the oddball. So now it would be not expected, but it would be unexpected. And then there is also oddball. So why not? Let's just add them all. And then we have condition, we have the 8. And for the 8, it's the same. It's also unexpected, but it's not an oddball, it's noise. So I'm just going to signal noise. So I have my HED tags. Oddball, nothing, expected, etc. So these describe in much more detail my event, because now if someone else do a similar experiment, they would use very similar HED tag, hopefully the same ones. So I need to validate them. Okay, no issue found. Then finish. It's telling me, okay, this is how your HED description is. So it's okay with me. I don't need to save the file. Now it's telling me the study, there's BIDS information in the study that have HED. So do you want to copy that in the study? Yes. And now tagging is complete. So we're going to export one last time. BIDS export free now has HED tags. And you want to resave the dataset? Yes. Okay, it's been exported, BIDS export free. We can always go online, double check that this is still valid. BIDS export free. It has one warning, which is about the HED string, but probably because we didn't put any event in for the ignore condition. But it's just a warning, so we can just continue and replace the dataset. So this is the level of description you want for your data. This way it can be fully reduced by others. And before I end this presentation, I also want to mention that there is, in the BIDS plugin, there is a file that contains an example for these. So that's the example 2. And it contains the full example of how to export these files. These are actually the same one I used here. I just used the 4 first subjects. And then also there's different blocks in the experiments. Each subject was doing 3 blocks. So you can see them here, 1, 2, 3. So there's 3 files per subjects. We only exported 1 here because it was just an example. We can also enter some notes. So I invite you to look at this script. You can do everything through script. The script is even better because, so here you see the description of the subject, and here the description of the participants, description of the columns of the participant, description for the events. Also, when we export from the command line, we can also rename the types. So these are all the variables we will give as input to the BIDS export. And the BIDS export will create this folder. It's actually the same function that's used from the graphic interface, but this time it's used from the command line. So thank you for your attention. And also, I want to mention one last thing is here, if you use the command line, you can actually export in BDF, because that's one of the accepted BIDS format. .set EEG lab files, when you use the GUI, it is going to export as .set files. So it's going to be the same raw data, but it's going to be in .set format. If you use the GUI, you don't have the choice. You have to export as .set. But if you use the command line, you can export in the native format as long as it's supported by BIDS. And BIDS only support 4 types of formats: EEG lab file, BDF file, EDF files, and Brain Vision Exchange files. If you use the GUI, they're always going to be converted as EEG lab dataset. So that's it for this short introduction. I hope you can now easily export your data. Thank you for your attention, and I hope to see you in one of my future videos.